So, good evening, guys. Today we will be starting with the extra class for the biochemistry course from NPTL by Professor K. Subramaniam. So, in this lecture, we will be discussing about the concepts that have been taught in the biochemistry course. So, this is an additional class. So, let's start. This is the first question for today. Use the reaction given below to solve the following question. So, this is a reaction in which palmitoyl coenzyme A is actually being oxidized and to carbon dioxide and water, and we have to use this reaction to find out how much water in kg can be produced from 1 kg of tripalmitoyl glycerol. The molecular weight of tripalmitoyl glycerol is 855, and ignore any water that may be produced by oxidation of glycerol. So how can we solve this question? Okay, so first we have to check what is tripalmitoyl glycerol. So this is basically a triglyceride. Okay, so that means it will have a glycerol. So this is a glycerol molecule, and it will be linked to the palmitic acid. Okay, so this is the structure of tripalmitoyl glycerol. Okay, so it's a triglyceride. So one molecule of tripalmitoyl glycerol will have 3 moles of palmitic acid. Okay, so that means if we say 1 mole of tripalmitoyl glycerol will have 3 moles of palmitic acid. Okay, so that means the 855 gram of TPG, that is the molecular weight of TPG, that will be equal to 3 moles of palmitic acid. And if we talk about 1000 grams, that means 1 kg of TPG, that will contain these many moles of TPG. Sorry, P, palmitic acid. Okay. Now, when these many moles of palmitic acid will go for oxidation, that means it will produce. So, 1 mole of palmitic acid produces 23 molecules of water. Okay, or 23 moles of water. And that means how much weight? 18 gram, because 18 gram is the molecular weight of water. So 23 into 18 gram of water is produced when 1 mole of palmitic acid is oxidized. Okay. Now, when 3000 by 855 palmitic acid moles are oxidized, that will produce 18 grams of water. Okay, so this much gram of water will be produced after these many moles of palmitic acid will be oxidized. And how these many moles? So because we have 1000 grams or 1 kg of tripalmitoyl glycerol. Okay, so after solving this, this would be equivalent to 1.45 kg. Okay, so this would be the correct answer. So option A would be the correct answer for the question. Okay, no, I hope the explanation is clear for the question. So let's move on to the next question. An amino acid has a non-ionizable R group. Okay, the pKa for the NH2 group is 9.4 and the COH group is 2.8. So consider the following statement. So there is an amino acid that have COH, NH2, the non-ionizable R group, and last the H. So this is the amino acid molecule. pKa for NH2 group is 9.4, for COOH is 2.8. Okay, so first we'll calculate the pi, that is isoelectric point of the amino acid. So that will be pK1 plus pK2 by 2. Okay, so this would give equal to 6.1. So this will be the pi. So what will happen at the pi? At pi, all the molecules would be sorry, all the amino acid molecules that we have would have a net charge 0. Okay, so at that means that if we put that particular amino acid in any of the electronic solution, it will not move to any of the poles. Okay, so let's see the options now. At pH 6, 6.1, 50% of the amino acid molecules will migrate towards the cathode when placed between two electrodes. No, this is false, because at pH 6.1, that will be the pi of the amino acid. At that pH, the net charge on the protein would be 0, and if we put any of the molecules in between the electrodes, then none of them will move any on either side. Okay, at a pH of 2.8, 50% of the amino acid molecules in solution are in the zwitterion form. Okay, so zwitterion form is the ionic form of this particular amino acid. That would be NH3 plus C, this is R, COO negative, and the H. Okay, so this will be the zwitterion form of the amino acid. So at pH 2.8, that means that is the pKa for COH. Okay, so 50% of the molecule will be in zwitterion form. Yes, this is correct. Okay, so at any of the pK values of the amino acids, what will happen? 50% of the molecules will be zwitterion, and 50% of the molecules will be ionized, positively charged. That means. Okay, so so option Q is correct. The pi of the amino acid is 6.1. Yes, this is correct. Just now we calculated. So option R is also correct. On titration of amino acid solution with NaOH, the amino group is deprotonated before the carboxylic group. No. So first, the carboxylic group will be deprotonated because the pH of this particular pK of this particular group is more, sorry, is less. So that's why whenever there is any slight increase in the pH, that would affect the ionic uh, characteristics of the COH group first, then the NH2 group. So this option is false. So which a pair of the above statements is correct? So in that case, option Q and R are correct. So A option is the correct answer. Okay. So let's move on to the next question. A cell with a mutation that inactivates phosphofructokinase PFK1 would likely exhibit which of the following? So first we check where phosphofructokinase 1 is actually involved. So PFK1 is involved in glycolysis. Okay, so now we'll see the options. So it will show, so it's a mutation that inactivates phosphofructokinase 1. So it will show increased rate of glycolysis and ATP production. So as it is inactivated, so it will not show the increased rate of glycolysis or ATP production. Okay, so this is false. Decreased rate of glycolysis and ATP production, but increased fatty acid oxidation. Yes, this can be the correct answer. Increased rate of gluconeogenesis and glycogenolysis. So this is not the correct answer. No significant change in carbohydrate metabolism. Uh, this will also be not the correct answer. So the correct answer is option number B. Okay, and as we can see here, phosphofructokinase 1 is actually involved in the conversion of fructose 6 phosphate to fructose 1,6 biphosphate. So it's a reaction because it is not reversible. That means it is actually being the reaction that is actually regulated by different methods. Okay, so it's an important reaction of the glycolytic pathway. So if this happens, that this enzyme is not functional, that means the glycolytic pathway will be stopped. Okay, and there will be significant change in the carbohydrate metabolism. And secondly, because this pathway is actually stopping glucose from being oxidized, that means there will be accumulation of glucose. So glycogenesis would be increased, not glycogenolysis. Okay, and the system, because the energy production from glucose will be stopped, that means the energy production from fatty acid oxidation will be increased. Okay, so option B would be the correct answer. Okay, so let's move on to the next question. Which of the following describes the role of chylomicrons? So they transport cholesterol from the liver to peripheral tissues. They transport dietary triglyceride from the intestine to the peripheral tissues. They are synthesized in the liver and transport endogenous triglycerides, or they are high density lipoproteins involved in the reverse cholesterol transport. So the correct answer is option B. And let's see. So chylomicrons, as we have studied before, also they are synthesized in the intestine. Okay, so they are not synthesized in the liver; they are synthesized in the intestine. Now, what happens? The lipoprotein lipase actually cleaves these and form the free fatty acids. Okay, and from there they are transported to different tissues where uh, uh, and yes, from there they are they are actually digested in those capillaries. Okay, and then the other cholesterol transporting units like HDL, VLDL, LDL will be transported to liver. Okay, so chylomicrons are actually being used to transport triglycerides basically, not cholesterol. Cholesterol is transported in the form of HDL, LDL, and VLDL. Okay, so this is actually a complex of triglyceride with the that is being transported to peripheral tissues. Okay, now let's see the options again. So the transport of cholesterol from liver to the peripheral tissues, no. Okay, so they transport from intestine, not from liver. So this is false. They transport dietary triglycerides from the intestine to the peripheral tissues. Yes, this is correct. They are synthesized in the liver. No, they are synthesized in the intestine. Okay. Now they have high density lipoproteins. No. Chylomicrons are not linked with the cholesterol form. Okay, cholesterol forming units basically. So the correct answer is option number B. Okay, so let's move on to the next question. Which of the following best describes the role of urea cycle? The options are: it synthesizes urea from ammonia and carbon dioxide, it breaks down urea from ammonia and carbon dioxide, it converts ammonia to uric acid, or is involved in the synthesis of creatine. Okay, so the correct answer is option A. It synthesizes urea from ammonia to carbon dioxide. Okay, so let's see. So as we can see, this is the diagram for urea cycle, and the first step is the formation of carbamoyl phosphate. And carbamoyl phosphate is actually being formed when ammonia is reacted with bicarbonate ions, or that comes from the CO2. Okay, now this uh, because we are not able to actually being use this ammonia as a secretion product. So we after this reaction, urea is finally removed from the body. Okay, so this helps in formation of urea from ammonia and carbon dioxide. Okay, so as the first option states that it synthesizes urea from ammonia and carbon dioxide, this is the correct answer. Okay, so let's move on to the next question. Which of the following conditions would lead to increased activity of the citric acid cycle? So the options are option A, high ATP to ADP ratio, option B, high NADH to NAD plus ratio, high levels of acetyl coenzyme A, all of the above. Okay, so we have to check increased activity of the citric acid cycle. Okay, so citric acid cycle is basically a catabolic pathway. Okay, so it actually helps in releasing carbon dioxide and water. Okay, so whenever ATP would be high, that means there would be no need of the citric acid cycle, so it will be inhibited. Okay, so the first option is wrong. Then if we have high levels of NADH, that means body has sufficient energy and it will also inhibit the citric acid cycle. Okay, when we have high levels of acetyl coenzyme A, so yes, when we have high levels of acetyl coenzyme A, so this actually activates or increases the activity of the citric acid cycle enzymes. Okay, both ATP and NADH are the negative regulators of the enzymes involved in citric acid cycle. Okay, but acetyl coenzyme A is a positive regulator. So whenever acetyl coenzyme A is increased, this would increase the activity of the citric acid cycle. Okay, so the correct answer is option number C. Okay, so let's move on to the next question. The Krebs cycle directly generates a small amount of ATP. What is the primary role of the Krebs cycle in terms of energy production? To directly power cellular functions with ATP, to capture energy released during breakdown in the form of high energy electron carriers NADH and FADH2, to completely oxidize glucose to carbon dioxide and water, to create a proton gradient across the inner mitochondrial membrane. Okay, because it produces a small amount of ATP. That means it will not be able to power the cellular functions with ATP. Okay, so this would not be the primary role of Krebs cycle. Then to capture energy released during the breakdown in the form of high energy electron carriers NADH and FADH2. Yes. So after the Krebs cycle is performed, the FADH2 and NADH are formed and they move to the ETC. Okay, for the production of ATP. So Krebs cycle actually helps the body to store energy in the form of NADH and FADH2. Okay, sorry. To completely oxidize glucose to carbon dioxide and water? No. Okay. Then to create a proton gradient across the inner mitochondrial membrane, this is done by ETC mainly, not Krebs cycle. Okay, so the correct answer is option number B. And here is the Krebs cycle. So it generally helps in formation of the NADH and the FADH molecule, and now they'll move to ETC for the proton gradient generation and ATP production. Okay, so this is the primary role of Krebs cycle, and the secondary role would be for the formation of CO2 and H2O from the compounds that have been formed in the form of acetyl coenzyme A. Okay, so let's move on to the next question. Based on the given Lineweaver-Burke plot for a reaction catalyzed by enzyme A, choose the correct statements. So this is a Lineweaver-Burke plot that has been given for a reaction that is catalyzed by enzyme A. So here is the question. Based on the given Lineweaver-Burke plot for a reaction catalyzed by enzyme A, choose the correct statements. So what can be the correct answer? So as this is a Lineweaver-Burke plot, so from the analysis, what we'll see first, because this is 1 by substrate on the x-axis. So this point would be 1 by Km, minus 1 by Km. So Km of the reaction would be zero point 0.1 millimolar. Okay. Then this point would be 1 by Vmax. Okay. So this is zero point 0.5. So that means Vmax is equal to 2 millimolar per minute. Okay, now the slope of the line would give Km by Vmax. Okay, so let's see the option. So Vmax is 2 millimolar per minute, Vmax is 4 millimolar per minute. So as we have calculated, Vmax is 2 millimolar per minute. So option A is correct. Now, if we have given ET, that is the total enzyme concentration as 2 millimolar, so we have got Vmax. So if we can see, because we have to find out the turnover rate, so we have to find out Kcat for that. So Kcat is equal to total enzyme into Vmax. Okay, so total enzyme we have 2 millimolar and Vmax was 2 millimolar per minute. Okay, so this would be 4 millimolar square per minute. Okay, now the turnover number is equal to 1 by Kcat. Okay, so this would be Sorry, I made a mistake here. So just a minute. So Vmax is equal to Kcat into total enzyme concentration. Okay, so we have to find out Kcat, that will be uh, Vmax by Et. Okay, so Vmax was 2 millimolar per minute and the Et was 2 millimolar. So that would give us 1 minute inverse. Okay, and the turnover number is basically the Kcat. Okay, so the turnover number is 1 millimolar, 1 per millimolar. Sorry, 1 per minute. Okay, so the correct answer is A and D. Okay, so Km would be 10 millimolar, not zero point 0.1 millimolar. So the correct answer is A and D. Okay, so let's move on to the next question. Calculate the standard free energy change for a reaction at equilibrium where the concentration of the product is 4 times that the reactants. Note that the reaction occurs at physiological temperature. Okay, so that means the product concentration is equal to 4 times the reactant concentration. So we have to find out the K equilibrium, that is product upon reactant. So that is 4R by R equal to 4. Okay, so if we have to calculate the delta G naught, that is minus RT ln K equilibrium. Okay, so R in this case would be 8.314, T at physiological temperature, that is 25 degrees Celsius or 298 Kelvin. So this is 298 Kelvin and natural logarithm of 4. Okay, after solving this question, this would come around minus 3.4 kilojoule per mole. Okay, so the correct answer is minus 3.4 kilojoule per Okay, so let's move on to the next question. Calculate the free energy change for the following reaction. So pyruvate is dehydrogenated, pyruvate plus NADH give lactate to NAD plus. Okay, so this is a reaction that is controlled by lactate dehydrogenase, and the concentration of NADH was 1 molar, NAD plus 0.1 molar, pyruvate 1 molar, lactate zero point 0.1 molar, and E naught is given for pyruvate, that is the reduction potential basically. So this is minus 0.185, and for NADH it's minus 0.320. So we have to calculate the delta G naught for the reaction. So first we have to calculate the E naught cell, that is the reduction potential of the particular reaction. Okay, so because in the reaction the pyruvate is actually reduced to lactate and NADH is oxidized to NAD plus. So the reduced one would be the lactate and the oxidized one would be the NADH. So this would be the for the reduced one, and this one would be for the oxidized one. Okay. So after solving this, it would come 0.135 volt. And to calculate this, Okay, so this is the formula for calculation for delta G naught from E naught cell. So this is minus F E naught cell. So delta G naught is equal to minus 2, because here we are having two electrons. So 2, Faraday constant is 96500. E naught cell just we calculated as 0.135 volt. So after solving this, this would come around minus 26 kilojoule per mole. Okay, so the answer can be in the range from 25 to 7, minus 27 to minus 25.7. Okay, so this would be the answer for the particular question. Okay, so let's move on to the next question. If three NADH molecule enter the electron transport chain, how many protons will complex 1 pump into the intermembrane space? Okay, so complex 1 is NADH dehydrogenase. Okay, so one NADH dehydrogenase basically pumps 4H plus to the intermembrane space. Okay, so as per the known number. So if we have from one NADH that is being dehydrogenated, 4H plus are being passed. So from three NADH molecules, it would be 12H plus. Okay, so the correct answer is 12, and here is the NADH oxidoreductase complex 1 basically. So it helps in the transport of 4H plus from to the intermembrane space from from the matrix of the mitochondria. And here is NADH that is dehydrogenated. Okay, so if this is from the one NADH molecule, then from 3 NADH it would be 12H plus. So 3 into 4 directly. So the correct answer would be 12. Okay. So let's move on to the next question. How many molecules of glyceraldehyde 3-phosphate will enter the regeneration step for 12 molecules of CO2 entering the Calvin pathway? So glyceraldehyde 3-phosphate is an intermediate that is formed from the uh, in the Calvin cycle. Okay, so at the third step, we have to take place in which regeneration takes place for the RUBP, that is ribulose 1,5 biphosphate. Okay, so as we can see here, when three molecules of carbon dioxide actually enters the pathway, six phosphoglycerate molecules are formed from three ribulose 1,5 biphosphate. So total number of carbon. This is a 5 carbon compound and we have three molecules, that means 15 carbon, and 15 plus 3 here, so that means 18 carbon. So now this is a 3 carbon compound, so this total would be 18 carbons. Okay, so from this, 6 molecules of 1,3 biphosphoglycerate are formed, then 6 molecules of glyceraldehyde 3 phosphate and dihydroxyacetone phosphate are formed. From here, one molecule of glyceraldehyde 3 phosphate actually enters the sucrose forming pathway. Okay, in plants. Now, 5 molecules of glyceraldehyde 3 phosphate actually enters the regeneration step for formation of the RUBP. Okay, now, so if we have 12 molecules of CO2 entering this pathway, okay, that means for 3 CO2 molecules, 5 molecules of glyceraldehyde 3 phosphate actually enters the regeneration step. For 12 molecules of CO2, it would be 20 molecules of glyceraldehyde 3 phosphate that will be entering the regeneration step. Okay, for every 3 molecules of CO2, 1 molecule of glyceraldehyde 3 phosphate enters the sucrose forming pathway. So, the other way around, if we see, for 12 molecules, it would be 4 molecules of glyceraldehyde 3 phosphate that will enter the the sucrose forming pathway. Okay, so 20 would be the correct answer. Okay, so this was the last question for today. I hope you guys have enjoyed the lecture series. So, thank you all for joining in, and let's close the session for today. All the best for your exams.